Hello, Sir EJ. Good morning po. My name is Merly S. Todabia from Section BAT 1D and also my partner, Judy Ann. We are here in front of you in the video to discuss the assigned topics that you given to us, which is the Module 4, Animal Nutrition, Lesson Number 1, Introduction of Animals.
Good day, sir. My name is Judy Andy Sabado from Bat 1D.
In this module, me and Judy Ann are discuss the Module 4 Animal Nutrition Lesson Number 1 Introduction of Animal Nutrition. The learning outcomes discuss the basic terms in animal nutrition and contrasting concepts of proximate analysis and gross chemical composition of plant and animal. Introduction. This lesson will bring you to the different basic processes of how nutrients are being measured and utilized. Nutrition is defined as series of methods by which organism takes and assimilates food to promote growth and replace worn out or injured tissues. Understanding the concepts on how to determine nutrient content, chemical reaction, and body utilization will help us further manage our bodies. The learning guide question. Number 1. What is the proximate analysis and gross chemical composition of plant and animals? The definition of terms. What is nutrition? Nutrition is a refers to the study of various physical and chemical processes that are transform food elements into a body elements. It is involves ingestion, digestion, and absorption of various nutrients from food. Nutrition encompasses several fields of discipline like biochemistry, chemistry, physiology, endocrinology, microbiology, and genetics. Nutrients are substances of elements found that are very necessary to support the animal's processes. These are carbohydrates, fats, proteins, vitamins, minerals, and water. Food feed is a term of given to any material that embraces both plants and animals, including the byproducts prepared from them. Example of the food feed is corn, rice bran, and fish meal. Feedstuff. The term refers to any stuff both prepared synthetically or natural and origin when properly used provide nutritional value in the diet. There are examples of feedstuff. Example is corn, DL-methionine, feed supplements, some vitamin premixes, and etc. Digestion. Digestion is the breaking down of feed particles into suitable products for absorption. May include mechanical forces, chemical action, and enzyme activity. Absorption. Transfer of a substances from the gastrointestinal tract GIT to the circulatory blood or metabolisms. It is refers to the combination of anabolic and catabolic reaction occurring in the body with energy liberation. And enzymes. It is refers to a complex protein produced in cells that will result in change in some substances within the body without changing its structures organic catalysis. Metabolic water refers to the water produced from the nutrients, carbohydrates, fats, and proteins when metabolized. Ration is defined as the animal's food with a weight nutrients balance for the animal's needs within the day feed requirement. Diet refers to the kind of food given to the animals regardless of whether and balance of nutrients requirements needed by the animals.
The gross chemical composition of plant and animal tissues. Proximate analysis. When we say proximate analysis, it is a set of laboratory procedures to quantity components of tissues and feeds. The different nutrients quantified in their corresponding methods are parameter description, sample preparation. Sample preparation is essential for the subsampling of the material before a determination. Dry matter analysis. Dry matter, by definition, is the part of the sample that remains after drying at 103 degrees Celsius. Crude ash. Crude ash, by definition, is a part of the sample that remains after incineration at 550 degrees Celsius. Ash insoluble in acid is the ash that remains after boiling in a strong acid. Crude protein. The term crude protein refers to the measuring the total nitrogen content and measuring the protein content using the conversion factor of NX6.25. Two ways the Cadal and Damas are available for nitrogen determination. Cadal method. Nitrogen is converted into ammonia absorbed in a boric acid and titrated against a standard acid. Damas method, with complete combustion of sample materials at 950 degrees Celsius within the presence of oxygen, nitrogen will be converted to a gaseous state and reduced to N2, followed by measurement in a thermal conductivity cell. Crude fat refers to the non-polar extractable fraction of the sample materials. Perform is extraction with or without prior acid hydrolysis, both being complementary methods. The laboratory should offer both options. Fiber analysis is done based on the samples boiling in a particular detergent solution and measurement of the remaining organic fraction. There are two methods available. Both are based on the digestion of feeds in detergent solution. Metabolism of feed directly in the detergent solution and filtration using crucibles. This is the official standard method. And digestion of the sample in a nylon bag, and to make it detergent free, washing was done to the bags containing the digested sample. Crucible based filtration method, digestion of feed directly in the detergent solution and filtration using crucibles. This is the official standard method. Nylon bag based method, digestion of sample while in a nylon bag and then washing it containing the digested sample to make it detergent free. Starch can be measured by the classical Ewers method or with an enzymatic process. The enzymatic method can be used for all sample types and is therefore preferable. Parameter description. Reducing sugars contain the essential sugars including glucose, fructose, and sucrose. The determination done is based on the Lafs-Schuler principle. Gross energy represents the total energy value of the sample and measured by bomb. Minerals, spectrometric methods following incineration and hydrolysis generally measure minerals. Amino acids, standard method for the determination of amino acids by a strong acid with or without previous oxidation, followed by chromatographic separation and detection after derivatization. Amino acids tryptophan, determination of tryptophan based on alkaline hydrolysis followed by chromatographic separation. Fatty acids, the standard method for fatty acids based on isolation and derivatization followed by gas chromatographic separation. Vitamins, determination of individual vitamins based on extraction followed by cleanup, concentration if needed, and chromatographic measurement. Mycotoxins. Mycotoxins are undesirable substances produced by fungi. This result in potential danger to human and animal body health. The different methods based on extraction, purification, chromatographic separation, and detection. Pesticides. Pesticides are undesirable substances whose maximum levels were defined in the regulation of national and international communities. These regulations demands a low detection limit and identification of the pesticides using mass spectrometric detection. The methods based on the derivatization, chromatographic separation, and identification. The next one are some common laboratory equipment used in approximate analysis. Oven drying. A drying oven is low temperature convection or forced air oven used primarily in laboratory settings. Specimens, tools, and temperature-sensitive chemicals are placed inside a drying oven to remove moisture slowly. And this picture is the example of oven drying. The second is ignition burning, muffle furnace where feed analysis for crude ash. Crude ash is by definition the part of the sample that remains after. This picture is the example of ignition burning. The third one is Cadle procedure, digestion, distillation, and titration. Below pictures is the example of Cadle procedure. The fourth one is ether extraction, sucklet apparatus. The last one is refluxing, digestion in acid and base ignition. And below the example picture. And that was the end of our report. Thank you for listening. God bless.